Hello, this is Bob Salter from Charm Sciences, and I'm excited today to present to you VerraGlo, a new technology for oil and fat oxidation testing. Today's talk, I'll talk about VerraGlo, which is a light-emitting diode chemistry, and a little bit about Charm Sciences as well. The light-emitting diode chemistry is essentially that. The chemical is exposed to the oil, and depending on the amount of hydroperoxide present in the oil, you get a linear luminescence output in about the 600 nanometer wavelength range. And you can see on this graphic, it's fairly independent of the oil type. And it's also a little temperature dependent. So if we can control the temperatures of the oils, we can get a nice coefficient of variation around each reading of peroxide values having to do with that particular oil in the ranges between 0 and 20 milliequivalents per kilogram oil. And as measured by the iodometric titration method. So you get a light output in a relative light unit relative to the PV value for oil. The method that Charm Sciences has developed with this chemistry is very simple. We make a tableted form of the enzyme, which is contained in a foil. And to start the assay, you pierce the foil, crush the tablet with a rod, and add your preheated oil, vortex for 10 seconds, thoroughly, screw on the adapter, put it in the Novalume, select the proper temperature channel, and start the count. And the Novalume will take that relative light unit emission and convert it into a micro equivalent per kilogram value. So the VerraGlo output is 1 PV of milliequivalent by the iodometric titration is equivalent to 1,000 of the VerraGlo output. And you can see with a variety of different oils at different PVs as measured by the iodometric method that you get a linear output. In this case, the highest value we've used is 12.9. But an R squared of 0.985, and I've blown up the area of more interest to the industry, which would be less than 4 PV and particularly less than 1 PV. The CVs that we see vary between 5 and 20% depending on what value of iodometric titration you're looking at. At the lower end, it tends to be a little more variable. This curve is at 30 degrees C, which would be basically for liquid oils, but for more solid fats, we might want to go up to 50 degrees C, for example, and run a curve at that temperature. The benefits of the method are obvious in that we're not really running a chemical extraction method. We're not using organic solvents. There's no toxicity or hazardous waste. It's performed in minutes, can be done by non-technical people in in a non-laboratory environment if necessary. The reagents are ready to use. And it's lower cost and less labor intensive and gives improved sensitivity at that lower PV range, or increased precision, if you will. The equipment required is an optional incubator if you need to get it to a controlled temperature, or if you know the temperature your oil is at, you wouldn't need an incubator. A steel rod to pierce the foil and crush the tablet, a positive displacement pipette and disposable tip for dispensing the oil into the sample, vortex mixer, an adapter to screw it onto the vial, and a luminometer where you would select the channel appropriate for the reading. It automatically waits the 30 seconds before it gives you a read. Our next steps, our next plans for this method are to start to receive some stakeholder input. We want to understand the temperatures that your processes are done at, develop curves for those processes, understand what your uses and specifications are for oil oxidation, select some trial locations where the method could be used for screening in comparison to a PV confirmation, and to work with the AOCS working groups in helping to have it become a more recognizable and accepted method in your organization. That obviously would include third-party laboratory validation compared to the iodometric titration, and ultimately collaborative study. Charm Sciences, I don't know if you're familiar with us. We are very active in the grain industry in making mycotoxin tests that are water-based extractions that have eliminated methanol and ethanol use in grain factories, and do provide online testing for trucks or a train car full of grains when they're received into grain elevators and milling locations. Charm is a US-based Massachusetts manufacturer of diagnostic tests. We've been in the industry focusing on food safety, process sanitation, quality diagnostic testing for 45 years. Our methods and the procedures that we do in our facility are customer driven, and we provide R&D development as well as technical support for all of our products. The luminometers that we use, you may be familiar, are being used for ATP sanitation hygiene verification, and Charm Sciences pocket swab is the most commonly used hygiene method in the United States today. There are thousands of luminometers in dairy food production facilities at this time. The luminometer is also used as a pasteurization indicator for milk in the dairy industry, and it has a ISO standardization as well as National Conference Interstate Milk Shipments approval, and it uses that same linear adjustment for RLU to make meaningful enzyme units, similar to the way we're doing it to the iodometric titration. Charm also makes simple to use microbial indicator tests, and our mission is to be committed to protecting our customers' brands and helping them provide the highest quality products and services to their customers. And I hope that you will, as part of AOCS, seek out Charm Sciences for anything that you might need. Thank you for your time and attention today. Again, my email, Robert Salter, is bobs@charm.com, and my title is Vice President of Regulatory Affairs and Business Development. Thank you again.